A patient monitoring system can display stable waveforms and numerical values even when the underlying signal conditions are no longer idle. In clinical environments, monitoring systems are expected to operate continuously and reliably. Once connected to the patient, they become the primary source of physiological information, supporting clinical observation and decision making. However, the values displayed on the screen are not direct measurements, they are the result of a complex acquisition and processing chain designed to detect extremely small physiological signals, amplify them, filter interference, and reconstruct meaningful clinical parameters. The reliability of this process depends entirely on the integrity of each stage in the measurement chain. For this reason, functional verification begins with controlled simulation of physiological signals. When verifying the ECG module, a patient simulator is connected through the limb leads and the precordial leads. The simulator generates electrical signals corresponding to defined physiological and pathological conditions, including normal sinus rhythm, atrial fibrillation, sinus tachycardia, ventricular fibrillation. This signal reproduce real cardiac electrical activity, typically in the millivolt range. The monitoring system must acquire the signal accurately, preserve waveform morphology, and calculate the correct heart rate. The waveform must remain stable without distortion or baseline instability, and alarm system must respond appropriately to simulated abnormal rhythms. This verification confirms the integrity of the entire signal acquisition chain, from the patient interface to the internal signal processing stages. When the monitor includes a recording function, waveform accuracy can also be confirmed through thermal printing. The printed trace must maintain correct amplitude calibration and time scaling, demonstrating that amplification, conversion, and processing stages operate correctly and consistently. Pulse oximetry verification follows a similar approach. The SpO₂ sensor is connected to a pulse oximeter analyzer, which simulates the optical properties of human tissue and pulsatile blood flow. The analyzer generates defined oxygen saturation levels and pulse rates, reproducing controlled physiological conditions. The monitoring system emits red and infrared light and measures variation in absorption caused by arterial pulsation. From this signal, oxygen saturation and pulse rate are calculated. By applying defined saturation and pulse values, it becomes possible to observe measurement stability, response time, and accuracy under controlled conditions. Non-invasive blood pressure measurements involve verification of both pneumatic and electronic subsystems. The pressure cuff is connected to a non-invasive blood pressure analyzer, which simulates oscillometric pressure signals corresponding to defined systolic and diastolic values. The monitor inflates the cuff, detects pressure oscillations, and calculates systolic, diastolic, and mean arterial pressure. Accurate pressure detection, stable inflation control, and correct signal interpretation are essential for reliable measurements. These verification procedures reveal a fundamental aspect of physiological monitoring. The monitor does not measure physiological parameters directly. It detects physical signals, often extremely small and sensitive to external conditions, and reconstructs physiological information through signal processing and interpretation. Each stage of this process must operate with precision. Electrical safety is equally essential. Monitoring system must maintain strict isolation between the patient and the main supply, ensuring that leakage currents remain within defined safety limits as required by international standards such as 6271. Verification confirms not only measurements accuracy but also signal integrity, isolation effectiveness, and system stability. When monitoring systems are evaluated under controlled and repeatable conditions, important differences in performance characteristics become visible. Signal stability, response time, and measurement robustness can vary depending on system design, signal processing architecture, and noise immunity. These characteristics may not always be apparent during routine operation, but they directly influence measurement reliability over time. A monitoring system is not simply a display. It is a precision measurement instrument designed to operate continuously, often in complex and dynamic clinical environments. Behind each waveform and numerical value, there is a complete engineering structure responsible for signal acquisition, protection, processing, and interpretation. Verification makes this structure observable. It allows the measurement chain to be evaluated, confirms that physiological signals are acquired and processed correctly, and ensures that the information presented reflects the true physiological condition. Ultimately, the reliability of patient monitoring does not depend on the presence of a waveform or a numerical value. It depends on the integrity, stability, and performance of the entire measurement system behind it. Engineering verification ensures that this integrity is maintained and that physiological monitoring remains a reliable foundation for clinical observation and care. Thank you for watching.